Hey, Michael. I hope he has your eyes.
Oh, what about your beautiful smile?
Hey, Mike. I hope our son doesn't inherit your life-altering Y chromosome link disorder.
Never mind that, Nancy. The baby's coming. Oh my god. What is that? It looks like the time-like dog motor possum, but worse. Oh, get it away.
Did someone say CRISPR-Cas9?
The clustered regularly into space short palindromic repeat, CRISPR, Cas9 system, consists of a nuclease known as Cas9 and a hybrid guide RNA, sgRNA. Originally identified in bacteria, type 2 CRISPR-Cas systems act as part of their adaptive immune response. Using a Cas9 nuclease protein, invading virus and plasmid DNA can be cleaved and rendered non-functional. The system allows for site-specific genetic modification through the introduction of a double-stranded break followed by the incorporation of a target sequence based on a supplied DNA template during the repair process. The protospacer adjacent motif, PAM, is a short DNA sequence directly adjacent to the sequence of DNA being targeted by the Cas9 nuclease. DNA fragments are taken up into the CRISPR loci, where they are transcribed and processed into cRNA. A cRNA, tracrRNA duplex is formed, which contains a guide sequence that is able to bind to target DNA. This in turn facilitates the action of the Cas9 protein, which produces a double-stranded cut in the target region. The addition of a guide plasmid, or single-strand DNA sequence, with a desired DNA sequence allows for Cas9-assisted homology-directed repair, thus resulting in integration of a chosen site-specific mutation. The applications that come with CRISPR are enormous, one of the most desirable being the genesis of designer babies for aesthetics and health benefits. Hooray! One of the major goals is using CRISPR to eliminate hereditary diseases from germlines. There have been several occurrences where it is shown this may be possible. Examples of these studies are the correction of hemostasis by correcting the human gene Y371D with the less severe gene Y371S, a cause of haemophilia B. Another great example is the correction of the gene expressing dystrophin, showing Duchenne muscular dystrophy is curable by the means of CRISPR-Cas9 system, with a correction of up to 62% success. Another example is sickle cell disease, which is found to have a success correction rate of up to 67.9% using the CRISPR-Cas9 system. Beta thalassemia is another disease that has been shown to be one of the many diseases possible to be removed from germlines. All these examples show the outstanding possibilities of the health benefits and possible aesthetic improvements available via the CRISPR system.
Wish we could afford CRISPR, but we's not very well off. But our baby is just going to be born pretty much as hideous as us, so...
Yeah, and I would never wish a life like ours upon our baby. One of ridicule and hatred by those upper society snobs. No job opportunities, no fit men.
Many claim withholding life-changing and life-saving technology possessing the ability to cure genetically acquired diseases is completely unethical. Others, however, claim that there is a lack of morality that arises in a scenario where eliminating genetic diseases has potential to result in complications in the future of altered germlines. While eradicating genetic diseases appears to be glamorous on paper, there is no predicting how the life of that particular genome will develop in the future. Another consideration is that genome editing is an extremely expensive technology that is most likely only affordable to those in rich countries, therefore placing them in an advantageous position when it comes to their children, furthering the distance between the high society and those in poverty. When editing genomes, the CRISPR-Cas9 technology has also been known to produce mutations in target cells, which can result in the death or transformation of cells. This occurs when the nuclease is overly productive and cleaves unintended sequences, or is inefficient and fails to do so. In December 2015, a summit on human gene editing called for a moratorium to be placed on biomedical research which involved edits to the human germline. But in recent times, non-viable human embryos by Chinese scientists have raised doubts about not only the ethics of embryo modification, but its efficiency, with only 14.3% of the embryos showing the desired mutations, and many more showing significant off-target editing. It is important to consider that all edits will be conserved, not only in the infected individual, but in their children.